Hi, my name is Liam Hunt. I'm an assistant professor at Rhodes College, which is located in Memphis, Tennessee in the US. And today I was going to talk about some research that I've been doing with undergraduate students here. We're a predominantly undergraduate institute, which is looking at this epigenetic phenomenon called position effect variegation and how we can use it to visualize age-dependent changes. So to start, I just wanted to mention this theory that was proposed where as cells age in an animal, they lose heterochromatin. Now, heterochromatin is the chromatin state which is more condensed and limits transcription. It's typically associated with histone modifications like methylation on specific lysine residues like lysine 9, and typically not associated with acetylation. And the heterochromatin loss model of aging suggests that in young age, cells have these heterochromatin enriched chromatin regions, which repress transcription and the appropriate histone modifications. But as the cells age, those histone modifications change. There's less methylation that represses transcription. There's more acetylation that enhances transcription. And overall, the chromatin state shifts towards something that's more euchromatin-like, where transcription is possible. Genes can be transcribed as well as repetitive elements or transposons like these ones here. And that repressed transcription is potentially quite detrimental because the cells have changed gene expression, they have altered functionality, and they can't maintain homeostasis as well because of it. So we were looking at this heterochromatin loss model of aging using a specific strain of flies that have something called position effect variegation. Position effect variegation basically refers to the position of a gene and whether or not that allows for transcription to occur in all cells. So in this case, it's dealing with the white gene in flies, this W+ here, which is normally located in a euchromatin region and readily transcribed. And when it is, it causes a red eye phenotype in the flies. Hermann Muller created these using his X-rays, created an X chromosome inversion in which the white gene was moved to a location that's closer to the centromere and subject to heterochromatin-mediated silencing in some cells. So the phenotype that we see there is essentially a mosaic of red and white, where the cells that show red in the eye essentially are expressing the white gene, and the cells that are white have heterochromatin-mediated silencing. So there's this mosaic expression. And we've used this particular phenotype to help understand how heterochromatin is formed. These flies were used where we took flies that were wild type and added in additional mutations of genes which could control heterochromatin formation. If loss of function of a gene that was important for heterochromatin formation occurred, you would typically see suppression of variegation, and those genes were called suvars. If loss of function mutation led to enhanced variegation, which would cause enhanced heterochromatin, so these genes were essentially opposing heterochromatin formation, these were called EVAR genes. And so essentially this eye phenotype can be indicative of an animal's ability overall to form and maintain heterochromatin. So what I wanted to test is whether or not this eye phenotype could actually be indicative of a heterochromatin-mediated loss that occurs in old age. And so what we'd expect to see there is the young flies have a relatively white eye phenotype, and then in comparison the older flies would have more red eyes, which would be associated with loss of heterochromatin-mediated silencing. And so to do that, we quantitate the eye phenotype in flies, and you can see examples of that in our white mutants, our wild type flies, or flies that have a white gene inserted into a euchromatin location. That eye color doesn't really change with age particularly much. When the white gene is present in a heterochromatin location, however, that eye phenotype does change. So as they become older, we do see more red eyes, which would be associated with a loss of heterochromatin that suppresses expression of that gene. And so the quantitation shows us something very similar in old age, 3 weeks of age. We get this mean grey value, which we quantitate from the eyes, which basically says that the eyes are darker. There's more red in them overall. And we can look at that progressively in individual animals, so longitudinally. And we do see, and I'm trying to highlight some areas here, like this one, where there doesn't really appear to be any expression of the white gene causing red pigmentation, but then as that animal gets older, we start to see the emergence of these cells expressing the red pigment due to the white gene. And so that suggests that as animals age, cells which otherwise did have heterochromatin-mediated silencing of the white gene have occasionally lost that. And to show that this coincides with transcriptional changes, we can measure mRNA of the white gene using qPCR. This is all qPCR measurements at the top here. And these PEV flies normally have suppressed expression of the white gene compared to a wild-type control due to its position effect. That's a young age. But then in old age, that's the red one here, there is increased transcription of the white gene. And over here, just some additional transgenic reporters that we've used that involve either HV-CYP or a LAC-Z gene, which is inserted into heterochromatin, and we have some euchromatin controls. And basically, we see in the heterochromatin inserts, there's increased transcription of those transgenic reporter genes. And if we were to look more broadly at the transcription of genes endogenously in flies, we can use RNA sequencing data. That's this here. Basically showing you whether or not there's an increase. So if these lines here go, if these bars go above the line, that means there's increased transcription. If they go below, that means there's decreased transcription. We see that there are some regions of increased transcription in old age flies compared to young age, and they tend to coincide with these heterochromatin enriched regions near a centromere, say for example of chromosome 2, chromosome 3, and the Y chromosome, which has a great deal of heterochromatin within it. So it suggests that not only do we see these heterochromatin loss in the phenotypes in the eyes, but also in the transcriptional profile of these genes that are located in heterochromatin. So if animals lose heterochromatin as they age, we were curious about whether animals which maintain better levels of heterochromatin could survive for longer. So luckily the PEV flies within a population are kind of heterogeneous in their phenotype. There's a big range of different phenotypes, from ones that have some really extremely enhanced variegation like this one here, through to ones that have very high amounts of pigmentation, so extremely low pigmentation compared to extremely high pigmentation, which would be associated with high levels of heterochromatin and low levels of heterochromatin respectively. So we can essentially sort flies on that phenotypical basis qualitatively, and then test how long they'll live for. And the ones that have low pigmentation that would be associated with higher amounts of heterochromatin tend to live longer than the ones that have high pigmentation that would be associated with low amounts of heterochromatin. And typically we look at the median lifespan here, which is about 15 days for the high pigmentation, closer to 20 days for the low pigmentation group. And in addition to them living longer, they seem to maintain muscle function for slightly longer in middle age as well. So in these flies, we measure climbing speed as an indicator of locomotor function, and that declines over this 3-week period. In the low group, though, that decline is a bit slower. So they appear to live longer and have a better health span if they have enhanced heterochromatin. So for this first part, we've been able to show you that age suppresses a PEV phenotype, which would basically be indicating that there is a loss of heterochromatin, which is associated with increased transcription. So we have essentially young healthy cells which maintain heterochromatin really well and have low levels of gene transcription due to that. But then in old age, that transcription increases, and that's due to a loss of heterochromatin. So to summarize the first part there, we've been able to show not just that there is this age-related loss of heterochromatin, that maybe the amount of heterochromatin being formed and their ability to maintain that heterochromatin could be indicative of how long an animal will live, which suggests that if we can find ways to improve heterochromatin function, we could use PEV phenotype as a guide, and this could indicate ways that we could slow aging. So we've tested this out using selective breeding. I mentioned that the population of flies is relatively heterogeneous. There's a big range from very enhanced to very suppressed variegation, but they average an eye phenotype that looks a bit like this here that I'm pointing to. If we, however, take those extremely enhanced ones and selectively breed them for multiple generations, we can get that average down to having very enhanced eye phenotypes, or we can selectively breed for the ones that have very suppressed phenotypes. So that would be associated with enhanced heterochromatin and suppressed heterochromatin levels. And if we test those flies that have been selectively bred for their lifespan, we find that in the males, the ones that have the enhanced or ENH selection live longer than the ones that have suppressed variegation in red here. And that's about a 20% increase in median lifespan. We don't see quite the same increase in females. These are non-fertilized females. So there may be a little bit of sexual dimorphism in this response. But we speculated that was due to the fact that we'd selected these flies on the basis of having enhanced heterochromatin. So if the reason they were living longer was because they had enhanced heterochromatin, which is what we selected them on the basis for, then we could introduce a mutation in a suppressor of variegation gene like SUVAR205, which encodes for a heterochromatin protein 1, or SUVAR39, which encodes for a histone methyltransferase that mediates the histone methylation necessary for heterochromatin formation. And if we introduce those mutations, we would lead to suppressed variegation, less suppressed heterochromatin formation, which we can see here, which should suppress the longevity increase that these ENH flies have. Yet, when we do that, they still appear to live relatively long. The ENH flies live longer than the SUP flies, even when we introduce these suppressor of variegation mutations, which suggests that heterochromatin suppression alone, having enhanced heterochromatin levels alone is not sufficient to promote longevity. And to further support that, we have a genetic manipulation here in which we have a duplication and an additional copy of the SUVAR39 gene, which encodes for that histone methyltransferase that will promote heterochromatin formation. And we can see phenotypically that it appears to do that based on PEV phenotype. But if we look at the lifespan of these flies, they don't appear to live any longer, which suggests that overall, just having enhanced heterochromatin phenotypes alone is insufficient to promote longevity. So there might be a little bit more to this enhanced heterochromatin allele than just enhancing heterochromatin to cause longevity. And in fact, if we looked at the transcriptional profile of these animals, so comparing our enhanced to our suppressed population, we don't see only transcriptional regulation in heterochromatin enriched regions. We see transcriptional regulation pretty broadly throughout all chromosomes, and there's a bit of transcriptional upregulation as well as downregulation, which suggests that our ENH allele, although it causes this enhanced heterochromatin-like phenotype, is probably also doing a lot more and is probably the reason why it's increasing longevity of these flies. So for this second part, what I've been able to show is that we can selectively breed and isolate genetic variants that have enhanced heterochromatin phenotypes, but that enhanced heterochromatin phenotype is not consistently associated with longevity. And the longevity phenotype is most likely a result of other effects that our ENH allele causes on these animals. And we are doing some studies to look into that a bit further, including looking at the physiology of these animals, their developmental rate, their size, their stress resistance. And for example, we can say that we have done some studies where we see that the ENH flies have increased resistance to nutrient stress, and suggest that those physiological changes could also be associated with their longevity. And we're also working on genetic complementation, which is allowing us to narrow down what specifically our ENH allele is that we've selectively bred for, which leads us to believe that it could be a loss of function mutation, specifically in a cytogenetic band within chromosome 2 that contains about 10 genes or so. So we're getting close to being able to narrow it down to one specific gene, and hopefully we'll be able to have a bit more information on that soon. But otherwise, what this data suggests overall is that, yes, there is a loss of heterochromatin that occurs as animals age, and enhanced heterochromatin could associate with longevity, but it's not strictly required for longevity. In fact, it may actually be that heterochromatin regulation is kind of secondary to factors that influence aging. And I can give you an example there. Reducing insulin signaling is well known to slow aging and increase longevity. We also show that it enhances heterochromatin phenotypes, but it may not actually be that reason that those animals live longer. It may be due to the alterations in protein metabolism that occurred when there's reduced insulin signaling that causes that age-dependent regulation. Right, so thank you all very much for listening. I also need to thank all of the following undergraduate students that contributed to the vast majority of this research. Really hardworking and fantastic students. So thank you to those students, and thank you to you for paying attention and listening. I appreciate it. Have a good day.